Welcome. My name is Kate McGrail, and I'm a member of the Community Health and Wellness Division at Suburban Hospital. We are pleased to have you here today for Unlocking the Clues in Blood, a new window into ALS detection. This is part of an ongoing series we have built with the Johns Hopkins Medicine Department of Cognitive Neurology. We thank you for engaging in the topics offered on brain health. We sincerely hope that you feel more empowered having spent time learning with our speakers. Today, we are turning our attention to a new topic, ALS. This development in unlocking clues about disease identification is fascinating in and of itself, but I know that for many of you in the room today, ALS has affected someone close to you. In this presentation, Johns Hopkins University neurologist Dr. Sonia Schultz and her NINDS colleague, Dr. Bolu Adewale, will explain their work and what it could mean for the future. They will also help us understand how this research fits into the larger effort to develop biomarkers, simple biological signs that can help diagnose and track disease like ALS and other neurodegenerative conditions. So let me share my slides. Well, at the bottom of your screen, there are a few icons that will help you today. We are already recording this session, and we'll share it to any who were registered and unable to log in. But at the bottom of your screen, there's a double C icon. So if you'd like to engage your closed captioning, please click that once. And because we're using Zoom webinar today, you will remain on mute. We highly encourage you to interact with our speakers, so please feel free to use the Q&A feature to type a question. Please note that the Q&A feature is different than the chat feature. We will reserve time at the end of the program to respond to your questions. And lastly, the program we offer is highly driven by your feedback. We kindly ask that you take a moment at the end of today's session to complete a short evaluation of today's program. A form will pop up at the end of the session when you hit the leave button. And if you're unable to complete the survey today, we will follow up with an email that will contain that link, as well as the recording. So now I have the honor of introducing our featured speakers. Dr. Sonia Schultz is a MD and PhD, a board-certified neurologist specializing in neurodegeneration. She serves as a senior investigator and chief of the Neurodegenerative Disease Research Section at the National Institute of Neurological Disorders and Stroke, NINDS, as part of the U.S. National Institutes of Health. In addition, she serves as an adjunct professor of neurology at Johns Hopkins University, where she contributes to specialized clinical care. Dr. Schultz is internationally recognized for her research on the genetic and molecular causes of neurodegenerative disorders, including Parkinson's disease, Lewy body dementia, amyotrophic lateral sclerosis, ALS, and rare neurological conditions. Her work integrates genomics and advanced proteomic technologies to better understand disease mechanisms, and identify biomarkers that may improve diagnosis, patient stratification, and future therapeutic development. A highly cited leader in the field, Dr. Schultz has published extensively in leading scientific and medical journals. Her contributions to neurology and translational research have been recognized with numerous honors, including the Normand Geschwin Prize in Behavioral Neurology, the Lasker Clinical Research Scholar Award, and the Presidential Early Career Award for Scientists and Engineers. And Dr. Adewale is a physician scientist whose research focuses on using data-driven approaches to better understand, diagnose, and treat complex neurodegenerative diseases. He received his medical degree from the University of Ibadan, Nigeria. He subsequently moved to the National Institutes of Neurological Disorders and Stroke, or NINDS, where he is currently a postdoctoral fellow. His work has led to the development of machine learning-based framework for the discovery of plasma biomarkers in Parkinsonism diseases. In recognition of his enthusiasm for genetic and neurodegenerative disease research, he was awarded the Sanger Prize by the Sanger Institute and received the Karl Dunesberg Fellowship from the Bayer Foundation. Through his work, he aims to translate multiomics data into biological insights for discovering novel biomarkers and therapeutic targets. And with that, we welcome Drs. Schultz and Adewale.
All right. Well, thank you so much for the kind introduction, Kate. So I'm going to get started and then Bolu will take over a little bit later. Let me just try to pull up our slides, and I hope you should be able to see the presentation right now, but please.
Yes.
Okay, well, fantastic. So we're going to talk about a research topic, which is focused on trying to detect neurological disease signatures in blood samples. And that is done with a very kind of pragmatic, idea in mind, because we want to detect disease easily, maybe in the primary care doctor's practice, maybe just by going to a local lab, without always needing access to subspecialty care. And so ALS is one of those very difficult to diagnose diseases early on. And so we're going to focus in on what we have done in this particular space. Okay, so before we get started, I just want to, show my disclosures here. As Kate has already told you, so we work for the U.S. National Institutes of Health. So everything that I tell you today is my personal opinion or my personal interpretation of the field. It's not necessarily the official position of the government. I do have some pharmacy sponsorship to work that is unrelated of what we're showing here. Relevant to today's talk is the fact that I have 2 patents pending based on using biomarker developments from plasma. And then Dr. Adewale is a fellow in my lab. He's an aspiring neurologist and training in modern technologies and how we can use them intelligently to give us insight into very challenging neurological diseases. He also is part of the patent that we have pending for Parkinson's disease. All right. So now let's dive into what we're going to discuss today. So I first want to get started on just giving a basic introduction of what we mean with ALS, or amyotrophic lateral sclerosis. What are the defining symptoms of the disease, and what are some of the challenges that we encounter in clinical practice in trying to diagnose the disease effectively. But also we'll comment on, current knowledge of possible risk factors that play a role, that give us some insights into the underlying biology. Now, there's still a lot that we don't know, but we continue to learn, and it's a very rapidly advancing field. And then I'm going to switch gears and specifically discuss what are biomarkers, and how does it, how are measures of disease that we can develop using fancy tools from, modern molecular technologies to imaging techniques, how we can use them effectively to diagnose diseases, but also tell us other things about the disease. Give a couple of interesting examples. And there's been a lot of development already in the broader neurodegenerative disease space. So for example, in Alzheimer's disease, we have very robust blood-based biomarkers available now that really can help us distinguish a patient with Alzheimer's disease from other forms of dementia. And it's really changing clinical practice quite rapidly. And so with that in mind, we actually wanted to, ask the question, well, can we detect ALS in the plasma as well? Are there signatures there? And so we combined 2 different type of advances. So one is called proteomics, and we're going to discuss a little bit what that means. So it's a measurement of multiple different type of proteins, building blocks in the blood, combined with a modern computational tool, and trying to identify fingerprints of the disease in the plasma samples. And then we'll discuss, based on our findings, what the biomarker, tells us, what are the opportunities, and what could that mean for the future. So let's get started and first talk a little bit about some of the names that you may hear a lot. So here in the United States, amyotrophic lateral sclerosis, or ALS, is often referred to as Lou Gehrig's disease, based on this famous New York Yankees baseball player who unfortunately, when he was quite young in his early 30s, actually developed a lot of motor control problems. So he started to stumble, he became weaker. There was a clear decline in his batting statistics, and there was some fatigue. So initially it was a little bit of an unspecific change, and people were initially suspecting that it may be lumbago, so compression maybe of nerves that could be going on. But the disease came out more and more, and subsequently he was diagnosed with amyotrophic lateral sclerosis, and really that brought this kind of disease to the attention of the public. Now, regrettably, he had to retire very young from his position, and sadly also then passed away in 1941. So he was quite young when he dealt with this particular disease. If you talk to individuals living outside of the United States, the disease is much more commonly known as Charcot's malady, named after the famous neurologist Jean-Martin Charcot, who lived in Paris in the late 19th century, and first described ALS with its core clinical features as having spasticity, which is an increase in muscle tone, but also weakness that really gets to the point that somebody is completely paralyzed. And so when he examined, patients with this condition, he noticed that there are two different problems. There is clear shrinkage of muscle, but there is also shrinkage of certain types of nerves that are associated with these muscles. And so the disease, amyotrophic lateral sclerosis, really defines those core clinical features. So amyotrophic, it just means there is a shrinkage of muscle tissue. And the reason why the muscle tissue is shrinking is because it doesn't get the signals that are coming from the brain all the way through the spinal cord and the nerves, so these so-called motor neurons. And the shrinkage of these nerve cells is also seen in the spinal cord, where those cells are located, the long kind of fiber tracts, and it's essentially describing the shrinkage of the spinal cord, which is the lateral sclerosis. So those are the core features. And to this day, we're actually looking for these core clinical hallmarks when we are diagnosing somebody with ALS. Now, so what we're essentially looking for is weakness or spasticity, usually a combination of those two due to impaired motor neural function. It can be very variable from one person to another as to how this disease usually first presents. So some people may have onset in their limbs, they may start to stumble, trip a little bit. Others have problems starting in their hands, where they may have difficulties opening jars, they may drop objects, etc. Some people start out with having symptoms in the so-called bulbar region, which is important for speaking and swallowing. So the speech may sound a bit garbled, like drunk sometimes is one of the descriptors. And subsequently, there can be involvement of muscles that also involve breathing. And that's really the major complication down the road, is that, breathing is increasingly impaired. We have learned over the years now that while the motor neurons are the most affected cell types in this disease, there's also a little bit of involvement of other nerve cells in the brain as well. So some people can also get some cognitive features, such as having difficulties with behavioral control, sometimes having difficulties with paying attention, with so-called executive function, planning, etc., and following through. It doesn't affect everybody. We don't fully understand why some people have these problems, other people don't. In some individuals, it can be quite pronounced, or it can be even the presenting problem. And then in these cases, we would speak about frontotemporal dementia as the clinical presentation of the disease. Um, it is, although it's considered to be rare, this is all relative. Across the United States, I mean, there's currently about 30,000 people dealing with this really serious disease. It's about, it's affecting individuals at various ages. The average age is about 60 years, but we have seen younger people as well. Lou Gehrig's is a classic example of a young onset case, and others who developed the disease much older than that. And there are different kind of variables and modifiers at play. So we don't fully understand all of those, and it's still part of ongoing research. So some may be genetics, some could be environmental risk factors, etc., that are playing a role. On average, there's about 5,000 people diagnosed in the United States every year. So it's a sizable patient population that are dealing with this really challenging disease. And it's not just, challenging because, it affects motor control. It's also challenging because it actually is relatively rapid in its nature and its progression. So the expectancy of survival from diagnosis to death can be 2 to 5 years. There is variability. We've seen people who survive much longer, and some people have a more aggressive disease course. We don't fully always understand it, but it poses a challenge because sometimes people, when they come to us, they're also already quite impaired. And so when we're starting to initiate treatments, there's already a lot of damage that has happened. And ideally, we want to catch patients very early on so that we can start the treatment. And as we're developing new therapies and new clinical trials, perhaps we want to get to a place where the disease doesn't become as disabling, or we can even develop the cures that are really very well needed. So it's labeled as a rare disease, but when you look for it, it's actually quite common. We see it quite a bit, and it has a considerable socioeconomic impact. So it's not an easy disease to deal with. So I want to just talk about a few known risk factors for ALS. So about 10% of all patients with this disease report a clear family history. And the other 90%, we label them as so-called sporadic cases, where the genetics is not playing as much of a role, but milder risk variants could still be somewhat involved. This is really a research area, but we have learned a fair bit about just focusing in on those familial cases, because we can study certain genetic variants in model systems. We can figure out what they do in a cell. We can try to recapitulate and fix it in the cell and then in an animal model, and then hopefully bring it back to the patient. So those studies are actually quite informative. There's also, very clear epidemiological evidence that people who have served in the military do have an increased risk for ALS. There may be multiple variables and explanations, such as being exposed to traumatic brain injuries, being exposed to chemicals, pesticides, etc. But we also know that, contact sports, traumatic brain injury, and excessive sports like running ultramarathons, etc., can also increase your risk for ALS, as is smoking, chemical exposures. Those all can play a role, but they only explain a little bit of the risk. For the most patients that we see in the clinic, we cannot tell you, why did you get the disease? For most people, we don't know. They are sporadic, and it's probably a combination of all of these factors. Maybe just some bad luck in addition, as well as aging itself. All of those can play together. So we call it multifactorial. I'm just going to focus in very briefly on some of the genetic milestones, because I think they have really given us a lot of clues as to what is happening. And so one of the tests that we like to send in the clinic very often is a genetic test for a certain type of spelling mistake in a stretch of DNA called chromosome 9. This is the most common cause of ALS and frontotemporal dementia in European ancestry individuals. We don't see it as much in Southeast Asians, so there are population differences, but particularly in Northern Europeans, we see this particular change. And so what's happening in these individuals is that, a naturally occurring kind of repeat signature in this particular gene called C9orf72. I apologize for this technical name. It's just the way we call it. It's, normally everybody has between 2 and 30 copies of that. And if it's in that range, it's perfectly fine. But for some reason, people with ALS, due to the spelling mistakes in these genes, the copy number gets repeated over and over and over again, so thousands of times. And so then it's really causing a lot of damage to the cells. And so we see it quite commonly. About 25 to 40% of all patients with a clear family history are found to have ALS due to this particular disease. And this is why a lot of the research studies are focusing in on the most common form of the disease. But not everybody who carries this particular spelling mistake necessarily gets the disease. There's a lot of variability. So some people, despite carrying it, and can age quite well and not ever develop the disease. And so there's modifiers at play. And it may explain why, even in those individuals where there isn't a clear family history, so-called sporadic ALS cases, we can also detect it in about 10% of individuals. And so there's things we still need to learn about this disease, about these modifiers, but we can already use it as a form of a test, at least in those individuals, as a biomarker in those individuals that have a clear family history. Now, there are other genetic forms. I'm not going to go into it. It's a long list of various acronyms that are confusing, but we are in the clinical practice. If we suspect that it could be familiar, we start to screen for the most common ones and those that have some clinical actionability. So there's another gene called SOD1, where there is an approved gene-targeted therapy available. It's a rare form of ALS. But if you have it, then you want to get on that therapy. So that's the genetic test is the way you can figure out that you have it. Okay. But the current challenge in ALS diagnosis is that a lot of patients, when they first develop disease, it is not clear as to what is going on. So some weakness may be attributed to some nerve injuries, maybe some back issues. And, common things being common, it's reasonable to rule those things out first. But the disease tends to be progressive, and then it becomes apparent that there is more than that going on. And so until a patient finally gets to a neurologist who has a trained eye and who really has the subspecialty expertise, a lot of time usually goes by. And it's on average 1 to 1.5 years that people have from first having problems to actually getting the diagnosis of the disease. And that is a problem because by the time they're getting diagnosed, a lot of things have already happened. And so we want to initiate the treatments as early as possible to protect those nerve cells, but we also want to get patients into clinical trials as early as possible to test if new drugs could actually help them. At the present time, we don't have a cure for ALS, but we are still working very eagerly towards a cure. And we need something to be more efficient and more specific in our ways of diagnosing patients with ALS. And so this is where the discussion of biomarkers is coming in. And so what we mean with biomarker, it's essentially a test that we do. It can be any kind of measurements, either of body fluids, such as blood, urine, and others, or another type of measurement, like an imaging study that tells us about something is going on within the body. It can be a measurement of health, it can be a measurement of disease. It can tell us something about how a person is responding to a certain treatment. It doesn't necessarily also always be associated with how somebody feels. So sometimes we can have measurements that we can take when somebody's actually still feeling fine, but it can tell us somebody it may be at high risk for developing a certain disease. And so there are good examples for that. So for example, measuring your blood glucose level, as shown here, for diabetics can be really informative as to how well your diabetes is controlled. if you, for example, go to your annual screening with your primary care doctor, there's usually a whole slew of different blood tests that are being sent off that check your liver function, check your renal function, check organs such as the thyroid, etc. And all of these give us interesting insight as to how the body is working, whether there is high risk for disease, or maybe there already is disease ongoing without necessarily causing any problems. And so we want to catch those early. We've also had some really exciting advances recently in the Alzheimer's disease field, where we have special type of brain scans, brain imaging, where we can detect certain proteins that are building up in the brain. And those are already used very successfully now in clinical practice to decide who should get novel therapies for Alzheimer's disease that can slow down the disease process. But there are different flavors of biomarkers as well. So for example, there's biomarkers that help us diagnose a disease. So for example, if somebody presents with chest pain, we send off so-called troponins to see if there is any damage to the heart cells, to the muscle cells in the heart. And if it's elevated and if you see changes in on EKG testing, we can diagnose somebody with a heart attack very quickly and efficiently. And so that's an example of a diagnostic biomarker test. Another example of a different type of biomarker would be a predictive test. So sometimes we want to know, well, which patient actually benefits from certain therapies, so we don't have to expose others who may not actually be good responders to possible side effects. And so, especially in oncology, there are really good examples of how that works. So certain types of breast cancer, for example, respond to targeted therapies. And this is really what precision medicine is about, trying to give the right kind of treatment to the right person at the right point of time. So that will be a predictive biomarker. And then there are so-called prognostic biomarkers. So sometimes, for example, cancer patients who have prostate cancer, you can look at how the tissue looks under a microscope, and then you can basically tell based on how aggressive it looks under the microscope, what the prognosis is. So that's important information for care planning, but obviously also for the patient and the family who are dealing with this disease. And finally, we can use it to monitor diseases. So it may not necessarily always tell us how to diagnose this, but we can see if our treatments are effective. So there's a lot of, for example, immunological diseases where we want to dampen inflammation, and we can measure inflammation over time and see how effective the treatment is actually working. And those are different ways of thinking. So not every biomarker is the same, but there is a real reason to think about this, because they can really elevate our abilities to do things and know about the disease in action. And so this is where we're coming in. We're thinking about, well, are there biomarkers that we can use that can help us diagnose the disease? So ALS is a neurological disease, and so it comes from the brain and the spinal cord. And so in order to detect disease in a very protected kind of environment, we have to try and think a little bit outside of the box. The body is doing a lot to try to keep it shielded from the rest of the organism, but thankfully, the brain and the other organs are still talking to each other. There's a lot of connection. And so this is where we can think about maybe we need certain type of fluids to analyze and see how the disease is ongoing. There's two fluids that neurologists in particular like to study. So one is the so-called cerebrospinal fluid, which is essentially a fluid that encases the entire brain and the spinal cord. It serves as a nice buffer. It is something that we can very easily access using a so-called lumbar puncture. It's not the most pleasant procedure, but for a neurologist, that's like a blood draw. It's pretty straightforward. But patients don't like it, understandably. You need a specialist who knows how to do this. There are some patients who can't get it easily because they may be on blood thinners, they may have had a lot of surgery, and it's not as easy to access. And so while the cerebrospinal fluid is really informative because it really is in direct contact with the brain, We're more interested in plasma as possible biomarkers because there's a lot of things that are changing with neurological disease that could also affect other organs, and we may be able to detect it. And so this is something where it's practically more easily to access. We can just take a blood draw, we can spin down the blood sample and then remove the cells and just look at what is left over, and this kind of yellow fluid that contains all of the proteins, etc. This is what the plasma is, and so this is really gold. This is where we can detect the disease, hopefully. And so that is the genesis of our research study. So can we detect ALS in the plasma? There's a little bit of research already ongoing to implicate biomarkers in ALS. So one biomarker that has been used and still is being used quite extensively in research studies is this marker called NFL, neurofilament light. It's essentially a protein that builds up whenever there is nerve cell injury occurring. Because nerve cell injury can occur in multiple different diseases, different contexts. It's not necessarily a test that can distinguish ALS from other diseases, but at least it can confirm that there is indeed a neurological injury pattern that is happening. And it has been used, for example, in clinical trials to show that certain interventions like the gene-targeted therapy for SOD1 that I mentioned, it can really dampen the nerve injury. So it tells us, that the treatment may be actually really working. And so it has been used to help track disease activity and treatment responses. But as I said, it's not enough to diagnose the disease. So that alone doesn't get us there. And so this is where our research study is coming in, where we have used plasma proteomics. So that's a much broader collection of proteins to study and a combination of them to develop a panel, a whole set of different proteins that serve essentially as a fingerprint of ALS. And so I'm going to hand the talk over now to Dr. Adevale, who has been specializing in these advanced analysis, and he's going to discuss the results of the study. And then we're going to, at the end, take questions and answers together. So over to you.
Thank you very much, Dr. Shows. Dr. Shows already mentioned the non-specificity of the NFL, in which we are able to tell that there is ongoing nerve cell injury or cell death, but we do not only need biomarkers that can tell us that there is ongoing neurodegeneration. We need biomarkers that can help us distinguish between ALS and other diseases that might also present with similar symptoms. So this was the basis for this study that was published in Nature Medicine last fall. And our job today is to summarize the findings from this study and the importance of our findings in ALS care and research. Just before I go ahead with presenting the findings, let's do a definition of basic terms. Some of us must have heard that proteins are often referred to as the building blocks of the body. There are about 100,000 proteins in the human body. And they make up important organs like muscles, nerve cells. Essentially, cells consist of these important proteins that serve important functions for our daily living and functioning. They are found in varying quantities in saliva, tear fluid, in the plasma, in the CSF, and so on and so forth. So the thing with proteins is that when we get ill, the levels of proteins in the CSF or in the plasma may be altered, that is, maybe increased more than normal or decreased than normal. And what we are able to do with proteomics, which you must have, a term you must have heard, is that we are able to make measurements of these proteins in large scale, that is about thousands of these proteins can be measured at the same time. And with these proteomic technologies, we can determine which specific proteins are either increased or decreased than normal. So there are several technologies or approaches that have been designed to measure proteins in the body. One of them is Olink, which was used in this study that I'll be sharing with you. So essentially, Olink uses antibodies to detect proteins. We are able to detect what is the quantity of the protein in the blood by estimating the number of antibodies or so-called probes that bind to these proteins. That is, the more antibody binding, the higher the level of the measured proteins. I also want to introduce a term many of us must have heard, artificial intelligence, which has become quite ubiquitous in our everyday lives. You probably can't imagine a day without ChatGPT and other AI tools. However, I'll be talking about a very specific flavor of AI known as supervised machine learning. And one way to understand this concept is to imagine teaching a child, or in this case, an AI tool, to be able to differentiate between a cat, a chicken, and a dog. Perhaps one approach you would use if you want to teach a child to be able to recognize this is to ask the following questions like, does it have a tail? If yes, it's either a cat or a dog. If no, it's a chicken. How many legs does it have? If two legs, it's a chicken. If four legs, it's a cat or dog. What is the first letter of this animal? If it's C, it's a cat or a chicken. If it's D, it's a dog. From the three questions I've asked, you probably would have noticed that you can make an absolute determination of what animal this is with just a single question. Essentially, you are able to determine what animal it is by combining all three questions together. For example, if you ask how many legs does it have, if you say four legs, it only tells you if it's either a cat or a dog. You need to ask a follow-up question to be able to determine if it's a cat or a dog. So, pardon my lame example, but as this kind of questions that I've asked in order to teach the AI, in this case, to recognize a cat, chicken, and a dog, are known as features. And these features are properties or attributes that are used to train the supervised machine learning model. So this process of teaching the AI model is called training, and the process of making predictions or learning how well the model has learned is called testing. So these are terms you would learn here from subsequent slides. So in terms of, you can also, in similar vein, you can compare this process of teaching an AI model to recognize a cat, chicken, and dog to also recognize ALS from controls. In this case, the features wouldn't be two legs or four legs. The features would be what quantity of protein do the blood sample have? What quantity of protein A does this blood sample have? What quantity of protein B? And you could also ask a following question that how old is the person with this sample? Or what is the sex? Or is this person male or female? Combining these features like I illustrated for the previous example, you can determine, our hypothesis was that, can we determine if a patient, a blood sample that came from someone, an individual, has ALS or not by combining these features or attributes that I just mentioned. So in order to do this, in order to test this hypothesis, we assembled people. There are three major categories of people involved in the study: people with ALS, people without ALS, but who had other neurodegenerative diseases that also have neurodegenerative processes. And we also had people who were apparently healthy, that is, had no ALS and had no neurodegenerative diseases. So we took blood samples from all of the study participants. Like I said, we divided this cohort into a training set that is for teaching the model to recognize ALS from non-ALS. And we also divided the other arm of this division was the testing set. We profiled all samples, all study participants, that is after collecting their blood, using the Olink proteomic technology I mentioned earlier. And then we conducted something called differential abundance analysis. Essentially, what this means is we are comparing the levels of all proteins that we measured in ALS cases comparing them to people without ALS. That is, we wanted to know the answer to the question, is the protein increased or decreased when you compare ALS to people without ALS? Next, the proteins that were most significant, with the most significant differences, were used as features, that is, the attributes that I mentioned, to teach or train the machine learning model to be able to identify ALS and distinguish them from non-ALS. So this was our approach to test this hypothesis. And what you see here is a graph showing the proteins that were altered in people in ALS when you compare them to people without ALS. You would see that we had about 5 proteins that were decreased in ALS, and you have about 28 proteins that were increased in people with ALS. You would notice at the top corner of this graph, you see NEFL or NFL, which Dr. Scholes mentioned earlier, that is associated with is neurofilament light chain that is associated with neurodegeneration or death or injury of nerve cells. This is not surprising to us, as it confirms previous research that have shown that NEFL tends to be elevated in people with ALS. This is the complete list of the 33 proteins that were either increased in levels or decreased in levels when you compare people with ALS with people without ALS. We further streamlined these 33 proteins and selected 17 proteins using methods known as feature selection, that is selecting the attributes, that is the proteins that best predicts ALS and best identify ALS. So if you recall this the concept of features, when you combine these 17 proteins with questions like, how old are you? Are you male or female? We can use the combination of these 17 proteins and these patient or individual attributes to determine with some degree of accuracy whether this blood sample came from a person with ALS or not. The graph on the right here is known as an AUC ROC curve. It illustrates the accuracy of this model or this AI tool that we have developed. You would see this diagonal line here corresponds to an AUC ROC of 50%. 50% is what you get if you were to guess at random whether someone had ALS or not. What you want in a very good test is a test in which the curve is almost at the top left-hand corner. And AUC-ROC curve is measured on a scale of 0 to 100%. A 100% AUC-ROC curve indicates a perfect test. And as you can see here, our AI tool was able to identify ALS cases with near perfect accuracy with an average AUC of 98.3%. There is in fact a large collection of samples in the UK, which was separate from what we have done in our lab, the samples we collected in our lab. So we decided to test this model on this collection of samples known as the UK Biobank. And we also found that the model, this AI tool we have developed, is able to differentiate between ALS and non-ALS cases with an AUC of 99.1%. Again, near perfect accuracy. We can also extend the capabilities of this tool to not only say whether one has ALS or not. Some of us are very number oriented and we might want to ask our doctor, on a scale of 0 to 100, what is my risk of ALS? We're very glad you asked, because this is another question that we attempted to answer in the lab. That is, using these same attributes, that is, these features, these 17 proteins and individual attributes, we are able to generate something called an ALS risk score on a scale of 0 to 100. And you would notice here on this graph that people with ALS, colored yellow here, they have a higher AUC ROC curve compared to people they have a higher ALS risk score compared to people without ALS. Again, you see a clear separation in the ALS risk score that is differentiating between people who have ALS and people who do not have ALS. This was replicated in samples collected in our lab and also samples from the UK. Importantly, we also want to be able to, like I mentioned earlier, differentiate not only between ALS and a healthy individual, which pretty much is doable for any physician, but we also want to be able to differentiate between ALS and similar conditions like neuropathies that affect the nerves or myopathy. These are conditions that affect the muscles. We want to be able to differentiate between these conditions that might mimic ALS. And in the UK sample, again, we see that of the 355 individuals who were diagnosed with either a neuropathy or a myopathy, the model, this AI tool which we have developed, is able to differentiate, say that with 93.8% accuracy, that these are non-ALS cases. This further demonstrates the capability of this tool to differentiate ALS from conditions that might confuse or that might be confused with ALS. Again, using the ALS risk score, which we've generated, we begin to see a fascinating story emerge from among the samples in the UK. For these samples in the UK, there were blood samples that were taken before people ever developed symptoms of ALS. So with the benefit of hindsight, we look back in time and we noticed that this score begins to increase gradually as one approaches the onset of symptoms in ALS. So apparently, these were people who would look apparently healthy without any symptoms of ALS. However, when you begin to evaluate using this AI model that we've developed in the lab, we begin to see that the ALS risk score, this quantifiable measure of risk, begins to increase as early as 10 years before they even develop symptoms. This shows that the ALS pathology or disease process is something that must have been going on for years before symptoms ever emerge. And it changes the way that we think about ALS significantly. Also, when you look at the proteins that are either elevated or decreased in ALS, we begin to see another pattern emerge, that we see these proteins are associated with functions like muscle development, the functioning of the nerve cells, and also maintaining homeostasis, that is a stable state in the body. We begin to see these alterations in these proteins in people who have ALS, showing that even years before the onset of the symptoms, before the patient ever complains of ALS, there have been compensatory mechanisms going on in the body to fight this ongoing death or injury of the nerve cells. This is this changes the way we think about ALS, in that it also presents a novel opportunity for us to intervene if we are able to say with some degree of accuracy who will eventually develop ALS or not. So the key findings, to summarize the key findings in this study, we found that plasma proteins, again, proteins are involved in functioning of the body. These proteins are altered in their level, that is, they are either increased or decreased. And the pattern we've seen with these proteins is that they're involved in functioning of the muscles, nerve cells, and energy metabolism. Also, we show from this study that using just 17 protein measurements in the blood, and also in addition with individual characteristics like age and sex, we are able to say with some degree of accuracy that this person would develop, either has ALS or not. We are also able to differentiate between ALS and similar conditions, not just differentiating between ALS and people who are apparently healthy. So this marks an important radical change in the field in the 150-year of history of ALS, in that it's like the first diagnostic biomarker, and we hope that we are able to take this further and develop this test further. So the work we've just described was carried out in the NIH in the Neurodegenerative Disease Research Section, led by Dr. Scholes and a collaborator, Dr. Brian Traynor. However, it's also important to know that this similar work like this is not just being done at the NIH. There are other labs also working to develop biomarkers for ALS. And also, there have been remarkable success which Dr. Scholes actually alluded to in the Alzheimer's field. Just last year, the FDA granted clearance for at least two blood-based tests for Alzheimer's disease. One of them was approved to aid diagnosis of Alzheimer's in specialty clinics, and there was a later test that was approved to help rule out Alzheimer's by primary care physicians. For ALS, we're not quite there, but we are quite closer than we were last year. And we've also published similar study for Parkinson's disease by combining machine learning and this large-scale measurement of proteins in the blood. So what does the future look like for biomarker research in ALS? We expect to make further progress to scale these tests and get it to the clinic, especially a non-specialist clinic. We expect it to shorten the diagnostic odyssey for patients who may wait months on months, considering the rapid progression of ALS, which is like 3 to 5 years. We expect this to buy patients more time and also help us to be able to intervene early. One of the problems with developing drugs for neurodegenerative diseases is that sometimes we get the right drug to the wrong patients because we are not so accurate at diagnosing. We expect this biomarker test to help us make accurate diagnoses, and in that way, we are able to direct the right drug to the right patients. And we are able to get better results from our clinical trials because the drug and the patients are well matched. So this research does not immediately translate to the clinic, but like I said, we are closer than we were a year ago. But what it has shown is that early detection and diagnosis of ALS is feasible. And even though there is no cure for ALS now, we are able to intervene possibly in the future to delay progression of this disease. So all of these research efforts is to gather enough molecular knowledge about the disease and to help us make accurate diagnoses. Effective therapy is our ultimate goal, and we believe that this biomarker-supported diagnosis will help us make better diagnoses, help us carry out better clinical trials that will eventually lead to better therapy. So it's been a pleasure telling you about this work that we've been doing in our lab. And these are some of the takeaways. We strongly believe that biomarkers are transforming ALS research. And we've been able to show through this research that we can accurately identify diseases at an early stage and possibly also intervene when we have better therapies. And we are looking forward to a future of precision medicine that we can have better diagnostic, better therapeutic options for patients. And this would help improve the lives of people living with ALS, their families, and their caregivers. So we've included some of these resources for your reading or study. And we also want to acknowledge our collaborators in the lab. We want to acknowledge our patients, study participants who have given their time, resources, and samples in contribution to ALS and other neurodegenerative disease research. So at this point, we would like to take questions, myself and Dr. Scholes. Any questions you may have for us? Thank you very much for listening.
Stop sharing. Okay.
So just a reminder, you won't be able to come off mute, so please type your questions in the Q&A section down below. Or if you're on an iPad or a tablet, it may be at the top of your screen. And we'll just give them a moment. But in the meantime, thank you both. Very fascinating work.
Well, thank you for the opportunity to present. It's always good to get outside of the lab every now and then, but there's a lot of development, so loads of hope for patients.
Absolutely. I am excited for you to get to the therapies stage.
That's the next
I've
target.
witnessed a family member enduring the decline, and we all know it's a very, very difficult thing.
Yeah. And I know this is a very challenging disease. And the rapid nature of it makes it so difficult to get the diagnosis and then start treatments. And so this is what we're working against. And I think there's real hope now that even preventative treatment trials could be feasible if we have something to measure the disease, detect the patients, and get the right treatments to them as soon as possible.
I'm curious about like what needs to exist for, say, payers to allow that to happen in a more like a common setting like PCP.
Yeah, I think there's a lot of things that still need to happen. So this is a research test. It works very nice in a well characterized study cohort in a very well informed environment where we can, essentially controlled for a lot of variables, technical variables. So the next test stage is to really see how generalizable is across different labs, across different settings, how people treated this blood samples, how often they have thought them, and how they process the sample. So we want to see how generalizable is across technical variables, but also across populations. So our study populations was very predominantly European ancestry. This is where we had the access to the largest collections of samples. And so there may be variables that we haven't yet attributed. So that needs to be developed in parallel. So we also want to make it ideally cheaper so that insurances may more likely actually cover the expense of the test. So right now, doing a proteome for the 3,000 fold proteins costs about $1,000. But we only need 17, so we are trying to develop a targeted panel just for ALS patients so that we can perhaps bring the price down to about $50, which would be tolerable. Maybe sometimes even some patients could cover it out of pockets because insurances sometimes lag behind. That's an ongoing battle. And then, there may be some, other proteins that we didn't detect as effectively using this pan. It's only a subsegment that we detect. There's other things we don't measure as effectively. There's some optimization that has to happen. But I think the first stages before it even gets rolled out as a clinical test for primary care doctor, that's the ultimate ambition, is we'll start using it in research studies. So we're already, apply it as a secondary outcome measure, so to speak, for clinical trials that are ongoing where plasma is collected and see how well it performs in these populations and whether it tells us already something that is already improving. Can we even on a molecular level measure that a certain therapy is actually working? So that's already a win-win. And as we learn more, we hope that we can get to the point that it can get approved as a clinical test. So there's a lot of things, but the research area will first pick it up.
Winnie is asking, are there any suggested sources for donations to be made to the cause?
So there's a lot of people who are working on ALS research. So the MDA, the ALS Association, very much engaged at NIH. We have the Foundation of the National Institutes of Health that are always very grateful for any donation. So there are loads of different foundations that are very much looking for additional research funding that can help advance these kind of efforts.
Wonderful. And my last question, since I don't see any others, is how do we stay informed for like recruitment to the studies? How do people learn about that?
Yeah. And that's a very good and very valid question. So some of the resources that Bolu actually listed on the last slide,
Okay.
such as clinicaltrials.gov, which is a large catalog that lists all of the ongoing clinical trials. That's a good resource. It's updated on a regular basis. But also the ALS Association, your local kind of multidisciplinary ALS clinic. There's usually researchers working in parallel with the MDA. As they're providing standard of care, they're also looking for study participants all the time. So those are the usual resources that I would point people to.
Oh, thank you. Thank you both. Thank you everyone for joining us today. I know your time is limited and your choices can sometimes seem endless. So thank you for carving out time to hear our wonderful dedicated speakers today, on behalf of Johns Hopkins Medicine, Suburban Hospital, and all the work being done literally across the street right now at the NIH. We thank you all. As you hit the leave button, you're going to get that evaluation. It takes no more than like 3 minutes. So if you could please fill that out, and I will follow up with you via email with a link to the recording and our slides today. So thank you, everyone. Thank you, Dr. Adewale and Dr. Schultz.
Thank you. It was a pleasure talking to you.
All right, take care. Thank you.
Bye-bye.
Bye.